For goodness sakes. Okay, so this is, do you know what, it might be easier to move the camera. There we go. A bit slippy, never mind. So this little video is going to be on aseptic technique. So you need to know why we use aseptic technique. Now, it is not to keep things sterile, because we want to grow microorganisms. That pretty much means where they're growing, it's not sterile. So what is it for then? So it's to prevent contamination of your cultures. So we are surrounded by bacteria. So, and if you're giving them their favourite food, you know, whatever lands on there will grow. And it is also to prevent what you're growing, your bacteria or your fungi, to prevent contamination of the environment. And that particularly applies, of course, to personnel. That's you. So that's why we do it. And there are some general things, some general points we can make about aseptic technique. So, we can make sure our equipment is sterile. And there are various ways of achieving that. Heat's very popular. So we just introduce you to the autoclave. An autoclave is a big pressure cooker. It heats to above 100 degrees centigrade, so it pretty much kills stuff. And we would use that for glassware. Can't really use it for a plastic petri dish, it'll melt. We do use it to actually dispose of our plates at the ends of experiments. We can use disinfectant. And this will be with particular regard to benches. And whenever you're doing microbiology, you're going to work close to a roaring or blue Bunsen burner flame. So what that heat does is it heads off towards the ceiling, spreads out over the ceiling, and it drops any bacteria that are in the air sort of out to the sides of you, so that you can work close to the flame, and you know all the air's going upwards and taking the microbes with it. And we can flame the necks of culture and agar tubes. And lastly, there was something else I thought of. Oh yeah, tilt the lids of petri dishes. When you're inserting agar or inoculating. So if you're going to put anything in your petri dish or take anything out of it, don't lift the lid right off, just tilt it. And there's something else that I just thought of then when I was talking about tilting lids. Lip's gone. Sorry. So we've got flaming. Anyway. Whatever, I'm sure it'll come to me and I'll add it on. So if we look at a little diagram of inoculation then, just to sort of show you. In this, we're going to take bacteria out of a broth culture. So this is our broth culture here. And we're going to either put it into another broth culture, or down this side, we're putting it into a petri dish. I think that's the thing I forgot, was to talk about not putting lids on benches. I'll do it now. So, An inoculating loop is a loop with a little metal handle, very fine metal, little loop at the end. It's really good for picking up bacteria. You can use it to pick up bacterial colonies if you're subculturing. You have to heat it to red hot. So obviously these are things that you sort of grab out of a jar, or you do in our laboratory, grab out of a jar. It's not sterile. To sterilise it you need to heat it in the top of a blue flame. This is our broth culture, so this is the culture with the bacteria in. You loosen the lid and you need to hold on to it, so a really good way of doing that is to hold it in the crook of your little finger so that it's not being put down on the bench in case it's got any bacteria on it and you don't want them on the bench. You then need to flame the neck of the bottle, and what that means is you pass it through very quickly through the blue flame with the Bunsen burner, and then you can insert your sterile loop. Now obviously it's important your sterile loop, you've just had it red hot in a Bunsen, that it's cooled down before you do that, or it kills everything. Importantly, most of your bacteria are in the bottle, so to prevent contamination of the environment you're going to stick the lid back on the bottle as quickly as possible. So you pass it through the neck of through the flame, put the lid back on, and then do your transfer with your inoculating loop. So if you're inoculating an agar plate, you get your agar plate with its lid on, nice and sterile, nothing growing on it, and then you're going to unsterilise it. So you're going to tilt the lid of the Petri dish, sweep the inoculating loop across, and wherever there are bacteria then we'll get colony growth. And then you seal it up doing the Holy Cross thing with the cross shape on the lid. If you're transferring it to a broth, then you need to do, so you'll have your inoculating loop in one hand, again unscrew the mouth of your bottle, flame it, put your inoculating loop in to transfer it into the sterile broth, flame, lid back on, and then re-sterilise your loop. So your used wire loop must be heated to kill any bacteria that remain. Now you need to be able to describe that procedure and these aseptic techniques. So I'm just going to add to that don't put lids on the bench. And certainly this is something that you should have done in your practical work in some form. You might have made a sterile plate and you will have done or been told to do all of these things while you were doing it. So that's sort of aseptic technique in a nutshell, and in my next video we're going to look at using it.